Hello, good morning, and welcome to today's webinar on Takara's next generation tools to clone, express, and purify proteins. Thank you very much for joining us this morning. My name is Malti Rahman, and I'm the Product Manager for Cloning and Protein Products at Takara Bio Europe, and I'll be presenting today's webinar. There will be time for questions at the end, but if you have any questions during the webinar, please just type them into the chat box. And if you have any problems with the sound or with seeing my screen, please also just type it into the chat box. Okay, thank you. Okay, so before I actually talk about the next generation technologies that we offer for cloning and protein expression and purification, I first just wanted to go through the typical clone, express, and purify workflow that most researchers use for their protein expression and purification experiments. So the first step usually involves cloning your favorite target gene. So this is the gene or protein that you want to study further into your protein expression vector of choice. Now most people or most researchers use traditional ligation-based cloning for this step, which can be time-consuming and laborious. And so today I'd like to present you our next generation infusion cloning technology, which we really believe is a better way to clone and allows you to obtain the right clone every time. So once the expression construct has been generated, the second step is then to express the target protein of interest in your expression system of choice. And in fact, at Takara, we offer a wide range of different protein expression systems, and I'll present a brief overview of the different systems that we offer and then go into detail about one or two of these systems because I won't have time to talk about all of them today. And then finally, once the target protein has been expressed, often with a tag such as a 6-his tag, the next step is then to purify the target protein. And if it's a his tag protein, you can normally purify it using traditional resin-based methods, which again can be time-consuming and laborious. And for this step, I'd like to present our next generation capture and membrane-based technology for his tag protein purification. Because really, this is a much faster and easier way to purify his tag proteins. Okay, so the first thing that most protein expression and purification researchers need to decide is which expression vector they want to clone their target gene into, i.e. which expression system they want to express their protein in. And in fact, there are a wide range of different systems that are on offer, and in fact that Takara also offer. So we have many different bacterial systems, including E. coli and bacillus systems. We also offer our baculoviral insect expression system, various different mammalian expression vectors, and finally we also offer an in vitro transcription and translation system, i.e. our human cell-free system. And as you can see, as you move from bacterial to human systems, the number of post-translational modifications that are possible increases. Okay, so now I just want to present this table which gives you a nice overview of all the different systems that we offer. So the first choice system that we always recommend to researchers is the E. coli PET Express and Purify system, which is simple to use and gives you a high yield of 1 gram per liter. And here protein expression is driven by the T7 promoter, and it's induced by IPTG. However, if this doesn't work and you have a more tricky protein which may be degraded by host proteases or which is not so soluble, you could also try our P-cold vectors or our cold shock expression system, where your target protein expression is driven by the cold shock promoter. So you induce protein expression at 15 degrees C. So this is ideal to increase the solubility of your protein because host protease activity is reduced at this temperature, and also you get more expression of your target protein compared to other host proteins at this lower temperature. And all these factors help you to obtain more intact and soluble protein. However, if you want to obtain very high yields of active secreted protein, you could, for example, use our bacillus system. So you could use either Brevibacillus or Bacillus. So Bacillus really is ideal for large-scale protein production, and you can even optimize secretion with our system. And in both cases, it's possible to obtain disulfide bond formation, and yeah, you get a very good yield of intact secreted protein. So in Brevibacillus, it's also good for toxic proteins. Finally, we also offer baculoviral insect expression systems. So this is good if, for example, you want more post-translational modifications and you want to retain a high yield. This is good, and it's also good for temperature-sensitive proteins. And then finally, if you have a very difficult toxic protein that you want to express in vitro, you could consider using our human cell-free system, which is an in vitro transcription and translation system. And our system has a higher yield than other available commercial systems. And then finally, one system that I didn't mention, but which is good if you have a very difficult protein that is just tricky to express, you could consider using our single protein production system. So here you alter all the ACA sequences within your target gene or protein, which enables you to just express just your protein using our system. So I won't go into the details today. You can find out more on our website. But this way you can really get high levels of your specific protein. So it's more labor intensive, but it can be good if all other systems fail. So in fact, the two systems that I'll cover in a bit more detail are the Brevibacillus and Bacillus systems today. Okay, so once you've chosen the expression vector that you want to clone your target gene into, the next step is to do the actual cloning step. And here I want to introduce you to our innovative next generation infusion cloning system, which we really believe is a better way to clone. So I know most of you will be aware that cloning usually represents the first step of most molecular biology experiments carried out today, but it's not usually the main focus of the experiment. It's rather the first step on to you to actually achieving your research goal, which in the end is to publish papers. So what if you had a much faster and more accurate cloning system than traditional ligation-based cloning? So we believe we have such a system with infusion. Imagine a cloning system that would allow you to clone both single and multiple inserts in a single step that you could also use for site-directed mutagenesis and to easily generate C-terminal and N-terminal vectors. Well, with infusion, you have such a system, a single system that can be used for multiple or all the cloning experiments within your lab. It's really a cloning system without any limitations. So you might be asking yourself, what's so great about infusion cloning? Well, the beauty of infusion lies in the fact that it's sequence independent. So it can really be used to clone any insert into any vector at any locus. So you're no longer restricted by available restriction sites. So you can be completely restriction site independent with infusion cloning. So this is a great advantage over traditional ligation-based cloning. And because you're not restricted by the sequence of the vector or the insert, you often don't need to do any subcloning. You can directly clone into your final destination vector, which is again another advantage over traditional ligation-based cloning. Infusion cloning is also seamless and directional. So this means that no extra bases are added to your insert unless you want to add them. So because you precisely define where your insert is cloned into your vector, there are no sequence scars that you sometimes can get with gateway recombination cloning due to the recombination sites, which can add extra amino acids sometimes to your target proteins of interest. But with infusion, you can avoid this. And because the cloning is also directional, you often need to screen less colonies before obtaining your positive clones. It's also possible to do nice applications such as multiple fragment cloning within a single step. So you can really clone multiple fragments in a single step, or do site-directed mutagenesis, including substitutions, deletions, or insertions in a single step with infusion. Okay, so let me now go into more detail of how this infusion system actually works. So the first step is to decide which vector you want to clone into, which can be any vector that you already have in the lab. The next step is then to linearize this vector, either through restriction digestion if you have suitable restriction sites, or by inverse PCR with a high fidelity polymerase if you want to be completely restriction site independent. The next step is then to generate your insert by PCR, which can again be any insert, but you simply need to use gene-specific primers that have 15 base pair overlap, shown in red and yellow here, which are homologous to the 15 bases found at either end of your linearized vector, also shown in red and yellow here. So these 15 bases will vary depending on which vector you use and how you linearize, but you simply just need to add the same 15 bases to either end of your insert, so that when you then incubate your purified insert and purified linearized vector with the infusion enzyme for 15 minutes at 50 degrees C, the infusion enzyme simply fuses the insert to the vector at these 15 base pair overlapping regions. And in this way, you don't actually digest the insert sequence at all, so you don't care about the sequence of the insert. So this is a common problem with ligation-based cloning, where you need to make sure that the restriction enzymes that you use to linearize a vector don't cut within the insert, whereas with the infusion you don't need to worry about this. It allows you to be completely sequence independent. So after the infusion cloning reaction has been set up, it's then simply transformed into E. coli, and it's within E. coli that you get any final gaps being repaired in vivo, and you get positive clones containing your recombinant construct. And the most important step for infusion cloning is the primer design step. So designing these primers correctly so that they contain the correct overlaps required for cloning into the linearized vector. And we have a new online primer design tool to help with this. So you can find this on our product page, and we developed this with Tesalogen. So now I just wanted to go into a little bit more detail on the exact mechanism of how the infusion enzyme works. So if you imagine, so this is the insert which has been generated with the 15 base pair overlaps found at either end, which are homologous to either end of the linearized vector. So the overlaps are shown in red and turquoise here. If you just imagine this is already linearized, this vector, so this black sequence is removed. What happens when you incubate the insert and vector with the infusion enzyme is that the infusion enzyme's 3' exonuclease activity in fact chews back the ends of the vector and at the ends of the insert, allowing the insert to spontaneously anneal to the vector at the exposed 15 base pair overlapping regions. And at this point, the annealed molecule is just held together by hydrogen bonds. It's only upon transformation into E. coli that any gaps are repaired, and you get permanent ligation of the insert to the vector happening in vivo in the cell, which then results in positive colonies containing your recombinant construct. So what are the primary advantages of infusion? So the biggest advantage is the high cloning efficiency. In fact, we achieve over 95% cloning efficiency across a wide range of insert sizes, so anything from 20 base pair oligos to 15 kb inserts. This is because we've invested over 10 years of progressive research into perfecting this product. And it allows you to clone directly into any vector, and the cloning is always seamless. And as I just said now, we have infusion has an excellent performance, over 95% positive clones. So really you just need to screen 2 clones per construct, and both clones should be positive. So this is a great advantage over ligation-based cloning, where often you need to screen at least 10 to 20 colonies per construct before you obtain your positive clones. Infusion is also fast and it's accurate. And it's also simple and powerful. So it can be used to clone large inserts, single and multiple inserts, also in a single step. It's really one system with multiple applications. So this graph here shows how infusion can be used to clone large inserts such as 8, 12 and 15 kb fragments. And as you can see, although the total number of colonies does drop as you clone larger and larger fragments, the percentage of positive clones doesn't change. So you can even clone up to 15 kb fragments quite easily with infusion. Now, this slide shows the accurate, directional and seamless nature of infusion cloning. So here we have sequencing data from several constructs that we generated with infusions. As you can see, there are no errors within the sequence due to the use of a high fidelity polymerase. The cloning is always directional, so you don't need to screen many colonies before obtaining your positive clones. And most importantly, the cloning is seamless, so there are no extra bases between the vector and the insert, which is particularly important when you're generating protein expression constructs because you don't want to add any additional amino acids to your protein, because potentially this can change the function of your protein. And in fact, infusion cloning is ideal for several applications. So not just single fragment cloning, but it can be used to clone into large vectors such as cosmid vectors. It can be used for single-step multiple fragment cloning, as well as deletion, insertion, and substitution mutagenesis. And it can also be used to add small N or C terminal tags. Really, we believe that you can design any construct, and infusion can be used to deliver this construct. Okay, so I'm going to just talk about the multiple fragment cloning and substitution mutagenesis applications of infusion, because these two applications are really nice applications of infusion. So how can you clone multiple fragments in a single reaction using infusion? So the first step is to, again, prepare a linearized vector. So this can be done either through restriction enzyme digestion or via inverse PCR. The next step is to generate your various different inserts, your multiple inserts via PCR. Okay, but using primers which either have a 20 base pair overlap to one end of your linearized vector or to the adjacent fragment that it will be joined to. Okay, so for example here, this example shows 3 fragments that will be cloned using infusion. Fragment 1 is shown in red, fragment 2 in blue, and fragment 3 in green. As you can see, the forward primer of the first fragment has a 15 base pair overlap with one end of the vector, whereas the reverse primer of the first fragment has a 20 base pair overlap with the second fragment shown in blue here. And the reverse primer of the second fragment also has a 20 base pair overlap with a third fragment. And finally, the reverse primer of the third fragment has a 20 base pair overlap with the other end of the linearized vector. And here primer design is very critical. And again, you can use our new online tool to easily design these multiple fragment cloning primers. And the reason we recommend using 20 base pair overlaps for multiple fragment cloning reactions is because this results in more colonies when you're doing multiple fragment cloning, because remember, as you clone more and more fragments, you're putting more pressure on your cells, so the number of colonies usually drops. So we have found, or our R&D has found, that using 20 base pair overlaps increases the number of colonies for multiple fragment cloning, whereas there's no real added benefit for single insert cloning, which is why we recommend 15 base pair overlaps for single insert cloning, but 20 base pair overlaps for multiple insert cloning. And then finally, when you have your various inserts amplified and carrying the appropriate 20 base pair overlaps, you again incubate the various inserts and linearized vector with the infusion enzyme, which effectively creates single-stranded regions at the ends of each insert and vector, allowing them to anneal at the 20 base pair overlapping regions. So in this way, all 3 fragments are effectively cloned as a single fragment into this vector using infusion. And this is a really nice application because you're not limited by available restriction sites. You're not limited by the sequence of the different inserts, because if you try to do this via ligation-based cloning, you'll often hit issues with available sites, and you would have to do several rounds of sub-cloning to generate this type of construct. And don't just take our word for it. We have a paper that used infusion for this purpose, and it's shown here. And in fact, we've just published online a specific multiple fragment cloning protocol using infusion. So you can access this specific protocol for this application on our website. Okay, now I just wanted to show you some in-house data that we have for 3-fragment cloning using infusion. So here we try to clone MBP Prof12 and ACGFP into linearized PUC19 using infusion to generate a final construct of 5.2 kb. And you can see here that after the infusion reactions, we obtain 89 colonies in total and only 1 colony on the negative control, which is good. And when 26 out of the 89 were screened further by colony PCR and also mini-prep and sequencing, you can see that 26 out of 26 were positive. So it's a really high efficiency even for multiple fragment cloning. Another great application of infusion is in fact mutagenesis. So this can be achieved by combining the power of inverse PCR with infusion. So how does this work? So if you imagine that you have a vector with an insert already cloned into it and you just want to make a specific substitution, shown in yellow here, you then simply amplify the whole vector via inverse PCR. But the inverse PCR primers at their 5' end need to carry the bases to be mutated, shown as X's here, but also will have a 15 base pair overlap with each other, so that when you end up with the final inverse PCR product, i.e. the linearized vectors and inverse PCR product, where the ends will contain the bases to be mutated and the overlaps with each other, you can then simply use infusion to recircularize this vector at these overlapping regions. And it's a similar principle for deletions or insertion mutagenesis too. And you can read more about this on our Nature Methods app note, so the link is shown here. And also I wanted to say that, again, our new online primer design tool can help with the design of these mutagenesis primers, which is often tricky to do. So this is made easy with our new online tool. Okay, finally, I just wanted to talk about our latest version of Infusion called Infusion HD Cloning Plus, which we believe really is the cloning solution, because inside the box you not only get the Infusion HD enzyme, but you also get a high-fidelity CloneAmp Hi-Fi PCR premix, which has a very low error rate, making it ideal for cloning, and it's also very fast. It has an extension speed of 5 seconds per kb. Our Plus kits also come with an insert purification kit or cloning enhancer to purify your DNA, and our stellar competence cells, which really work best with infusion. So the reason we provide this complete solution is because it contains all the optimized subcomponents required for successful infusion cloning. But equally, outside the box, if you have problems with your cloning reactions, we also provide expert technical support, and we're always trying to improve our products, and we're dedicated to our customer success. And in fact, this Plus system is a value packaging, because trying to purchase these items separately would work out much more expensive. Now, so once you've cloned your expression construct, the next step is to express the target protein. So here, as I explained before, we have many different types of expression systems, but the two I'll concentrate on today are the Brevibacillus and Bacillus systems. So the Brevibacillus system allows you to obtain secreted protein expression. And in fact, we have two different options now. So if you prefer that the protein stays within the cell, you can use the vector which doesn't carry the secretion signal, i.e. the intracellular vector. And the reason Brevibacillus is good is it has a very low level of protease, so you get active secreted protein, or intracellular if you prefer. And also it's endotoxin free, which is unlike E. coli. So for many proteins, it's better than E. coli, and you get a higher yield. It's also good because it allows disulfide bond formation, so it's good for expressing eukaryotic proteins. And also it's good for toxic proteins because if you use a secreted vector, it doesn't stay in the Brevibacillus cell for very long. And we have two different versions of our Brevibacillus system. So one with the classical shuttle vector, where you first need to clone in E. coli your target gene. You first do the cloning step in E. coli. The vector is then shuttled into Brevibacillus afterwards. Or you can use our new BIC system, where you can directly clone in Brevibacillus, so you don't need to use the shuttle vector. Instead of a two-step cloning, it's a one-step cloning. So it looks very similar to infusion, but it's a little bit different. So here you clone into our PBIC vector, which is provided as a linearized vector, and you generate your target gene so that it has 15 base pair overlaps to either end of the PBIC vector. But in this case, when you transform both the insert and vector into Brevibacillus competent cells, in fact, the cloning occurs via a homologous recombination in vivo. So it's not exactly the same as infusion, but it's easier than using the classical shuttle vector. So this is a nice system that you could consider using. And another nice system that we offer, which is quite unique, is our B-Satilla system, which allows you to optimize secretion of your protein. So B-Satillus is good for medium and large-scale fermentation studies because it's a safe organism. And with our system, actually you can generate a library of your specific target protein with different signal peptides. In fact, we provide 173 different types of signal peptide within our system. So when you have this library, you can then see which signal peptide leads to best or optimal secretion of your target protein, and then go ahead. So you can kind of screen different peptides for optimal secretion. Okay. So now, once you've expressed your target protein, so many of our systems include, that I've talked about just now, include his-tag vectors. So it allows you to express your target protein as a his-tag protein. The next step is then to purify the target protein. So most people purify his-tagged proteins using resin-based methods, which can be very time-consuming and laborious and sometimes expensive. And often you need to do a lot of optimization for each protein. And so here I'd like to introduce our next generation CaptureM membrane-based purification systems, which allows a much faster and easier purification of his-tagged proteins. Okay, and in fact we have CaptureM kits that allow both purification of his-tagged proteins as well as untagged antibodies. Okay, and in both cases we offer mini prep, maxi prep and 96-well formats of our kits, which are shown in this picture here. So in the case of our his-tagged purification kits, these columns or plates contain a high-capacity nickel-containing membrane, which allows his-tag protein purification. Whereas for antibody purification, these columns contain a high-capacity membrane with protein A, and protein A is able to bind the Fc portion of antibodies, thus allowing antibody purification. But the great thing about our CaptureM technology is that it allows a really fast and easy purification, which is completely resin-free. So the purification takes just 5 to 15 minutes, depending on whether you use the mini prep, maxi prep, or 96-well formats. So the mini prep can be completed within 5 minutes, and the maxi and 96-well within 15 minutes. And the whole protocol can be done at room temperature. And as you can see, the protocol is very simple. It's literally equilibrate your column, bind clarified lysate, wash, and elute. It's as easy as a plasmid mini prep, you could say. And the good thing about our CaptureM membrane is it's compatible with many additives and lysis buffers, and because of the small bed volume, you end up with a very highly concentrated eluted protein. Okay, so you might be asking, what's so great about this capture membrane? What makes it so unique? So if we compare our capture membrane technology with traditional resin-based columns, you can see that resin-based columns look like this. They have a large bed volume, which enables high capacity, but because of the large bed volume, you have long separation times because of the slow diffusion of macromolecules within the pores of the resin. And in fact, we do have resin-based products such as His-60 nickel and Talon, which allow his-type protein purification. So you can get a good yield, but it takes a long time and it often needs to be optimized for each protein. However, with our Capture-M membrane-based technology, our Capture-M columns actually contain this high-capacity membrane. So here we've modified the membrane pores to enable high capacity. So traditional membrane-based columns have been plagued with low capacity because of the small pores, but because we've modified our membrane pores, it allows the columns to have a higher capacity. So in this way, you don't compromise on yield, but the purification is very fast due to the small bed volume, which allows a rapid flow through the column. So that's what makes it special. In fact, you can see this is what our capture membrane actually looks like. So first I'll talk about our His-tag protein purification kits. So this is the protocol, and as you can see, it's very simple. So whether you're using the mini prep 96 well plate or maxi prep format, the first step is to prepare clarified lysate, either from bacterial cells, or you can use mammalian cells, or you could even use cell culture supernatant directly. This is clarified to remove any debris which could clog up the column, and this is loaded onto an equilibrated mini prep or maxi prep column or 96 well plate. And this results in fact, immediate binding of the protein to the membrane. So this binding step often needs to be optimized when you're using resin-based columns, but without Capture-M membrane, this is immediate because it's so flat, and this is where you save time. So after the binding step, there's a wash, and then there's an elution step. And in fact, if your sample is very dilute, you can do several loads. So you can do several bindings before the wash and elution steps. Once you elute, if you analyze your eluate by SDS page, you can, we found that you get very clean bands, so very pure proteins coming through. And this is the typical yield that we obtain. So with a mini prep, it's 100 micrograms per column. With the 96 well plate, it's 100 micrograms of protein per well. And with the maxi prep, this goes up to 2.5 mg per column. So the only difference in the protocol is that the spin step is longer for the 96 well plate and maxi prep. So in between each step, you have a 3-minute spin, whereas it's 1 minute for the mini prep. So you can imagine with the mini prep system, you can purify your proteins within 5 minutes. So it's really very, very fast and very convenient. If you're screening many colonies, this is, or many protein construct, this could be very useful for you. And with the 96 well plate format, you can also use a vacuum manifold if you don't want to do a spin. And the other really good thing about our Capture-M membrane is it's very robust, so it can tolerate many additives such as EDTA or beta-mercaptoethanol or TCEP or DTT. So often EDTA can damage resins, which is why people do a buffer exchange after lysis. But because our Capture-M membrane can tolerate up to 10 millimolar EDTA, up to 30 millimolar beta-mercaptoethanol, up to 10 millimolar TCEP, and also up to 10 millimolar of DTT, it's very robust. So you don't need to do any buffer exchange prior to purification, because actually the protein doesn't sit on the membrane for that long because of the low bed volume, which is good because it's able to tolerate these additives. And we've also tested many other additives which are shown in this table, and you can find this table also online. Now, in fact, our Capture-M membrane can also be used to purify difficult proteins such as membrane proteins. So, for example, G-protein coupled receptors are membrane proteins which are hard to purify because of low expression levels, because of their high hydrophobicity, so they're not very soluble, and because they have a low stability in detergents. So how does our Capture-M membrane help researchers to overcome these three problems? Okay, let me explain that. So in terms of the low expression levels, often what researchers do to get around this problem is that they express the protein in a high volume to get a sufficient quantity of the target protein. But if you're using resin for this, often you need to use a high volume of resin, which takes a very long time, and it can end up being very expensive. But with our Capture-M columns, you can do several loads of dilute samples onto the column. So for example, if you use our MaxiPrep columns, you can do effectively 2 loads of a 200 ml culture. So in this way you can pass a high volume of lysate but still purify a concentrated protein because you elute in a small volume, whereas if you're using a high volume of resin you will still need to elute in a higher volume, and often you need to concentrate your sample afterwards, but this can be avoided using our Capture-M columns. In terms of high hydrophobicity, often researchers need to use detergents or other additives to make the protein soluble. Now, for example, with resin-based columns, using such detergents can actually damage the resin. So often researchers just add the detergent to the lysis buffer and then do a buffer exchange prior to purification with resin. But this adds time and cost to your experiment. Whereas with our Capture-M technology, there's no need for buffer exchange because the membrane is robust and allows you to use additives directly in your lysis buffer. And because the protein doesn't sit on the membrane for very long, it can tolerate these additives, and you get a concentrated protein at the end. And also, as I was just saying, another issue is that these GPCRs have a low stability in detergents, but because the Capture-M technology is so fast, you still get very good protein functionality after purification because the protein doesn't sit on the membrane for very long. So the protein time in the detergent solution is minimized to minutes, and this leads to improved reconstitution of the protein after purification. And don't just take our word for it. This is data that we have from a researcher who used our Capture-M mini prep columns to purify a GPCR. So he used it to purify a 6-his-tagged adrenoreceptor beta 2. So he expressed this his-tag protein in 293T cells and then prepared clarified lysate from these cells, which he then loaded onto a mini-prep column, carried out the mini-prep as per our protocol, and then after SDS page and blotting onto PVDF membrane, you can see that Ponshu staining of the membrane revealed this protein present in all the fractions, so it's this band here. When this membrane was then probed with the ADRB2 antibody, you can see that they were able to see the ADRB2 protein. They were able to see that it's concentrated in the eluate. So in this way, he was able to purify this GPCR within 5 minutes using our Capture-M mini prep kit. And similarly, we have Capture-M kits for antibody purification for those of you who do antibody expression and purification. The only difference is here the membrane has protein A immobilized within the membrane. So you have a high capacity protein A membrane, and protein A is able to bind to the FC region of antibodies. In terms of the protocol, it's very similar to the HisTag mini-prep protocol. The only difference is, of course, the starting sample, which will be an antibody sample. So here you can start either with cell supernatant containing your antibody, or serum, or asite fluid. So after loading this onto an equilibrated capture M protein A mini-prep column, again, you get immediate binding of the antibody to the membrane, and you can do several loads here. If the sample is dilute, then there's simply a wash and elution step. And you can see that when you do STS page with the eluted proteins, you get a very nice clean bands for the IgG heavy and light chains. And you can see that there's very little albumin coming through, which is often a problem when you're doing antibody purification. And again, in terms of timing, the whole mini-prep protocol can be completed within 5 minutes. And here you can see that our capture protein A mini-prep, or the membrane itself, is able to bind antibodies from various different species. So here we use sera from various different species, and you can see we get quite nice, clean purification from various different species. But the yield depends on the species. And also we tested some commonly used sample types, such as mouse IgG1 from hybridomas and human IgG1 from 293 or CHO cells, and we got fairly good yields here. But interestingly, a really nice application of our capturing protein A membrane is that it allows fast and efficient immunoprecipitation and co-immunoprecipitation. And in fact, we've recently launched our Capture IP and co-IP kit, which contains basically the Capture protein A mini-prep columns, as well as the various buffers required for immunoprecipitation. So how does this work? So the first step involves preparing lysate from your target cells, which is expressing the target protein that you want to immunoprecipitate. So here we try to do this with PP2A beta subunit, which was expressed in 293 T cells. We then used a specific polyclonal antibody against this protein to do we incubated the lysate with this antibody in a tube, and this tube was subjected to end-to-end rotation for 10 minutes at room temperature. This sample was then loaded onto an equilibrated protein A mini-prep column, and then the rest of the protein A mini-prep protocol was followed. And you can see within the eluted proteins, we were able to purify this PP2A B subunit. So this is a blot with a monoclonal antibody against this protein. So it's a really nice application. We were able to do this IP, you could say, within 15 minutes, which is really fast, because normally this can take several days to weeks, depending on whether you need to optimize your conditions or not, depending on your target protein. And similarly, you can also, okay, so sorry, I probably didn't explain. So how would protein A membrane allow this immunoprecipitation? This is because when you bind the sample at this step, the membrane will capture the antibody. So this is a PP2A antibody, but also will capture the target bound to the antibody, which is how you're able to do this immunoprecipitation. And similarly, you can also do co-IP this way. So here we prepared lysates from 293 T cells, which we know express P53 and SV40T antigen endogenously. So these are endogenous proteins that we co-IP'd. And we know that P53 and SV40T antigen are known strong interactors. Okay, so when we did the same as here, but for the co-IP, we either precipitated with the SV40 polyclonal antibody or the P53 polyclonal antibody. And here, at this binding step, you would get purification of, you would get capture of either the SV40 or P53 antibody by the membrane, but you would also co-purify the bound target and its interactors, so SV40 and P53 in this case. And as you can see, when we then did a blot with monoclonal antibodies against SV40 or P53, we could detect both SV40 P53 in 2932 cells, whether we IP'd with the immunoprecipitated with the SV40 antibody or the P53 antibody. But when we did the same experiment with HT1080 cells, which we know doesn't express endogenous SV40, we weren't able to immunoprecipitate these two proteins. And you can read more about this application on our tech note. Now, I just wanted to end this webinar by talking about our Capture-M trypsin kit, which will be coming out soon. So here we will be providing kits where trypsin is immobilized within the membrane, which allows you to do fast tryptic digests. So normally when you're doing mass spec analysis, you need to first digest your protein into peptides. And typically researchers do this in solution, and this normally involves an overnight incubation. Whereas with our Capture-M trypsin columns, this tryptic digest can be done in under 2 minutes. And when we compared the typical in-solution digest versus our Capture-M trypsin columns to digest apomyoglobin, you can see that 70% digestion was achieved with the in-solution methods, whereas we obtain 100% digestion with our Capture-M trypsin column. So it's really nice. And equally, with our Capture-M trypsin columns, you can use a sample as it is. You don't need to denature, reduce, or alkylate it, as is usually the case when you're doing overnight in-solution digest. And again, we illustrate this with the myoglobin protein. So you can see this is the intact protein. When you do an in-solution digest with just the sample as it is, you get 0 digestion. Whereas with the Capture-M trypsin columns, you get 60% digestion, even with no denaturation. And finally, I just want to end with some nice data that we have, some nice mass spec data for apomyoglobin and whole cell lysate samples. So here we either tried to digest apomyoglobin with our Capture-M trypsin columns, and then when we did a downstream mass spec analysis of the peptides generated, you can see that we obtained 100% sequence coverage with the known sequence of apomyoglobin. And this is data from Dr. Bruning at the MSU. Similarly, Dr. Kian from USC, the University of Southern California, actually tested our Capture-M trypsin columns with whole cell lysate BT47 from these BT474 cells, which is a breast carcinoma cell line. And you can see that he was able to use our columns to generate peptides, and then he was able to identify more than 2,320 proteins within this whole cell lysate. So it's a nice proof of principle results, proof of principle for our Capture-M trypsin kit. So this will be coming out very soon. If for those of you who do mass spec work, this could be very useful for your experiments. Okay, if you have any more questions, you can always learn more about our products by visiting, for example, the Cloning Resource Portal, which has lots of information about our next generation infusion system. Or if you want to learn more about our protein products, including our Capture-M technology, you can go to the Protein Resource Portal. Or you can email us at techeu@takarabio.com. So I have time for some questions now, if anybody has any questions. In any case, all of you will receive a copy of this presentation. So if you have more questions later on, you can ask me. Okay. We just have one question. So is infusion similar to Gibson assembly from NEB? Yes, they are similar in the sense that both are seamless cloning technologies. which involve exonuclease activity. But actually, Gibson assembly also has a in vitro ligation step, and also the gaps are filled in by a DNA polymerase in vitro, which often leads to more errors, whereas this happens in vivo with infusion. So it happens within the cell where gaps are repaired and the permanent ligation occurs. So this leads to less background with infusion compared to Gibson. Okay, I also wanted to add that we have samples of both the capturum histagon protein A kits. So if any of you are interested in testing the technology, you can contact us about these samples. And similarly, we can offer a trial rebate on infusion if you're interested in testing the technology. Okay, thank you. We don't have any more questions, so I wish you all a nice day, and yeah, get in touch with us if you have any more questions. Okay, thank you.